So let's start the second part of chapter 23 by remembering our definitions of digestion. We have both mechanical and chemical digestion, and mechanical digestion is simply the mechanical breaking down of food into smaller pieces. The advantage of this is that it increases the surface area for enzymes. For example, if we have a piece of food that is like this, the enzymes can bind to these surfaces, but if we cut it in half, it can also bind to these surfaces. So that's the benefit of mechanical digestion. Again, chemical digestion is what uses those enzymes to literally break the chemical bonds, to have them undergo hydrolysis reactions, to make large molecules break into smaller molecules. This slide shows you an overview of the different kinds of digestion. With carbohydrate digestion, we start with starch and disaccharides. And as I mentioned before, within the mouth, we have salivary amylase. Remember, if it ends in ASE, it's an enzyme. AMYL is going to be addressing carbohydrates, so amylase. Salivary amylase is what starts the digestive process for carbohydrates. And then when that chyme enters the small intestine, pancreatic amylase is added to the mix. And this will take large sugars, oligo means many saccharides, and disaccharides, and begin to break them down into smaller and smaller disaccharides. Disaccharides cannot be absorbed into the body. So we will take a look at how those are handled by the brush border so that they can then be absorbed. We'll do that in another slide. Our overview of protein digestion, of course, starts with proteins. We do not see proteins handled in the mouth, but once it reaches the stomach, we have seen how pepsin is secreted by the gastric pits, and in the presence of hydrochloric acid, it works very well to break down large polypeptides, which then will enter the small intestine, and our pancreas, again, secretes pancreatic enzymes, proteases, trypsin, chymotrypsin, and carboxypeptidase, all to break those down in the small intestine into small peptides and a few small polypeptides. But again, it is not until they are exposed to the brush border enzymes that we see individual amino acids being cleaved off. It is at the amino acid level, some dipeptides and some tripeptides that can be absorbed. We will see those in detail again in another slide. Fat digestion and absorption is really pretty cool to me because it's different. Now, we do have some lingual lipase present on the tongue that just barely starts a little digestion of lipids. The gastric lipase in the stomach also is of fairly minor importance. Lipid digestion earnestly starts in the small intestine thanks to the contribution of bile salts. Bile salts emulsify fats. Emulsification of fats is not chemical digestion, but is mechanical digestion, taking a large fat globule putting bile salts on it to create many little fat globules. You've seen something similar to this when, say, you've had to clean up after turkey dinner at Thanksgiving. You put water in the bottom of the pan, and when you stop putting water in, you see a big fat globule float to the top. All you have to do is put a little detergent in there, and it will break it up into small fat globules. That is emulsification. Once fats are emulsified, they can then be exposed to the pancreatic lipases that are secreted from the pancreas into the small intestine. And at that point in time, triglycerides are broken down into monoglycerides and fatty acids. Now, although this slide does include nucleic acid digestion, I just don't cover it in this class. All right, let's take a closer look at carbohydrate digestion and then absorption. So here at the top, we see how our oligosaccharides, disaccharides are going to be broken further by pancreatic amylase. And our disaccharides then will be splashed up against the brush border. Within the brush border, there are membrane-bound brush border enzymes. Now, some of those brush border enzymes will be maltase. They break down maltose. We would also have sucrase. They will break down sucrose. And then finally lactase, which breaks down lactose. They have the ability to break these disaccharides down into monosaccharides. Once they are at the monosaccharide level, they can be absorbed. Let's take a look at that. Before we get into the mechanism of absorption of especially glucose, I want to talk about these really interesting cells that line the villi of the small intestine. They're interesting because they have very few mitochondria, only enough to meet the needs of the cell. Because there are few mitochondria in these cells, the concentration of glucose in these cells is fairly high. In other words, if there were mitochondria, the minute the glucose comes into the intracellular fluid, it would go into the mitochondria, and therefore the intracellular fluid concentration of glucose would be low. Without those mitochondria, there's a high concentration of glucose in the intracellular fluid. Well, that sets up a problem for us. And the problem is we have monosaccharides, glucose in particular, that needs to be absorbed into the cell against its gradient. It is high in the cell. Compared to the outside in the lumen, glucose is lower. We do not have an ATPase-driven glucose pump. It's just not there. We do have what is called an S-GLUT. I'll talk about that in a minute. On the basolateral side, we have our sodium-potassium pump. And then also on the basolateral side, we have a facilitated diffusion protein carrier. Let's look at this mechanism then. All right, so we are looking now at this mechanism. Again, this carrier is very specific. The textbook doesn't tell you this, but it is called an S-GLUT, sodium glucose transporter. It uses secondary active transport. You may have to reach back into A&PII Ch23 to remember this. What is the primary active transport? Well, it is our sodium-potassium pump that starts this whole cascade of activity. We do need a molecule of ATP to phosphorylate the sodium-potassium pump so that it can work. The sodium-potassium pump puts sodium out of the cell. And for that reason, the concentration of sodium in the cell is low. Remember, our concentration of glucose in the cell is high. So this primary active transport has created a sodium gradient so that it's low on the inside. Sodium is high in the lumen of the gut. We have a lot of salt in our food. And so, because it's low inside the cell, we can use a carrier protein without using any additional ATP. Sodium can bind to this carrier. And when it does, glucose will also bind to it. And without any additional ATP, the sodium concentration gradient drives this mechanism and brings glucose in against its gradient. So this is secondary active transport. What is the driver here? ATP. What is the driver here? The concentration gradient, specifically of sodium that was generated by this one. Okay? Let's look a little bit further at this. So, sticking with the apical inside of these cells, let me add one more thing. And that is that if our orange monosaccharide here is fructose, fructose has its own carrier protein. It does not need ATP. And so, well, in fact, let me see if I can draw one in as an orange one. The fructose would bind to this, facilitated diffusion, be brought into the cell, and then there is another one on the other side of the cell where it will be carried out into the interstitial fluid. It takes no ATP to absorb fructose. It at least takes one ATP to absorb glucose. And this is the theory behind the trouble with high fructose corn syrup products, adding weight and contributing to obesity in our population. It doesn't take any energy to absorb fructose. It does take a little energy to absorb glucose. One final comment here. On our basolateral side, as I said, we do have facilitated diffusion going on here with a carrier, protein carrier, that can take fructose and glucose across the membrane. It takes those products into the intracellular fluid, and from there, through bulk flow, those molecules can move into the capillary. Remember, we talked about bulk flow when we talked about how at the capillary level, net filtration happens, or net reabsorption. This slide takes us to the proteins and how they are finished being digested and then absorbed. So again, our pancreatic proteases can break the peptides down into small peptides. And those small peptides then are splashed against the brush border that has specific enzymes that are called aminopeptidases. And these aminopeptidases will cleave off individual amino acids from the peptide. These amino acids may also bind to these carriers I called S-glutes in the previous slide. And so the S-glute not only has a binding site for glucose, but there's a piece of an amino acid that resembles a piece of the glucose that can bind to the exact same spot. We think of this as a potential competition between the absorption of glucose and the absorption of amino acids. It doesn't happen that much across the intestinal wall, but we are going to see this concept in the kidney later on. So, why are amino acids transported in this way? Secondary active transport, because our sodium-potassium pump engaged in primary active transport and created the sodium gradient that drives our secondary. Once the amino acids are brought into the cell, they also are going to be taken out to the interstitial fluid with a facilitated diffusion mechanism, and from there it'll be picked up by the capillary through bulk flow. Now, fats are handled in a very different way. These small fat globules that are in the small intestine have been broken down. Large fat globules are emulsified, and then pancreatic lipase breaks them down even further. So these fat globules are emulsified by the bile salts, as you see here. The bile salts also kind of stick to the surface of these small globulates or droplets of fat. The pancreatic lipases then target those fats and break them down into monoglycerides and free fatty acids. Now, in addition to the bile salts, there is something called lecithin, which combines with the bile salts to form micelles. These are small structures that contain monoglycerides, free fatty acids, some cholesterol, and the micelles are in a position to cross the cells to go into the enterocytes of the small intestine. Now, once they go into the enterocytes, the bile salts and lecithin are removed, and you have free fatty acids and monoglycerides that are in the cell. Well, they are put back together, and they are packaged with other fatty substances and some proteins to form what are called chylomicrons. And it is the chylomicron then that leaves the enterocyte and goes into the interstitial fluid. From there, they will go into the lacteals. And the lacteals empty into lymphatic vessels that will take the fats back to the bloodstream by the lymphatic ducts emptying into the subclavian veins. So this is what I find so unique and interesting about fat absorption. In the lumen of the gut, the chemical digestion, breaking the fats, the triglycerides, down into free fatty acids and monoglyceride, that occurs in the lumen here. Those are packaged. They are brought into the enterocyte, where they're put back together again before they're fully absorbed into the body. That is so bizarre to me. Instead of keeping them at the monomer level, at the very smallest level, they are put back together in this step of the chylomicron. And it is in that form triglycerides end up getting absorbed into the lymph. I just think that's weird and really pretty cool. This is a big table, and you do not have to know it all, but what we are looking at is hormones that aid in digestion, and we've seen cholecystokinin being involved in fat digestion. We have seen GIP, glucose-dependent insulinotropic peptide, GIP, sometimes called gastric inhibitory peptide, that also responds to a fatty chyme. Gastrin increases hydrochloric acid secretion, stimulates gastric emptying, etc. All of these things. Now, we have not looked at histamine. We have not really looked at intestinal gastrin. We have looked at secretin. Take a look again at all of its activity. Other than that, we really have not looked at the various hormones here. So what do I want you to know? Secretin, gastrin, GIP, and cholecystokinin. In summary, the vast majority of the absorption of our nutrient molecules, which are all solutes, occur from the small intestine, the lumen of the small intestine, across these hepatocytes into the interstitial fluid, and from there into the capillary. If you're moving that many solutes across membranes, what will follow it? If you were thinking osmosis, you are correct. Increase the solute concentration here, you create an osmotic gradient, and water follows it. So I put table 23.2 in here at the end of part 2 so that you can again take a look at digestion and absorption through the different organ sections. Make sure that you're clear on everything that's in the notes. Make sure that you can identify it here in this slide.